The glio? That's what it's called?
Hello. Say hello.
Well,
Ruben and I are continuing with our study notes for life science and cell biology. We have reached the section, we just finished learning about diffusion and movement of molecules, etc. And as we conclude that section, we're gonna go over a few notes of a few details. So, one note is definition of glial cells, which we mentioned earlier as we were talking about the different kinds of cells, and we were talking about the cytoskeleton. So, this is showing cells in blue color, showing the chromosomes inside cells, and the cytoskeleton, the external cytoskeleton in green. And so different kinds of cells have different structures, even though they're very similar, but there's slight differences. So glial cells are defined, they surround neurons and they provide support for and insulation between neurons. So, glial cells are the most abundant cell types in the central nervous system. Types of glial cells, more specifically, oligodendrocytes, astrocytes, ependymal cells, Schwann cells, microglia, satellite cells. Glial cells are the glue of the nervous system, playing a critical role in the development and function of the brain. So there's some photos. Nervous system, so the central nervous system, showing oligodendrocytes, microglia, astrocytes, ependymal cells. So their structures are slightly different. Peripheral nervous system has Schwann cells, satellite cells. More pictures of what the cells look like. So their shape defines their function. So they're kind of cool. So central nervous system and peripheral. A diagram showing parts of these cells. So the dendrites extending out the cell body, or the soma, axon, hillock. So that's showing the neuron, and an animation, the nucleus in the middle. So we have axon, myelin sheath, axon terminals. It says a myelinated neuron. Dendrites receive electrical and chemical messages from other neurons. Cell body processes include signals and generating outgoing signals. Axon sends outgoing signals to axon terminals. So as a single glial cell wraps itself around an axon to form a segment of the myelin sheath. Axon terminals make contact with nearby cells and transmit signals to them. So information flows through the neurons. Okay, another note about desmosomes. Desmosomes are a part of the cytoskeleton we mentioned earlier. They are also known as macula adherens, plural adherentis, Latin for adhering spot. It's a cell structure specialized for cell-to-cell adhesion, structure by which two adjacent cells are attached, formed From protein plaques in the cell membranes linked by filaments, they are button-like spots around all cells. Therefore, desmosomes are intercellular junctions that provide strong adhesion between cells and tether intermediate filaments to the plasma membrane. Because they also link intracellularly to the intermediate filament cytoskeleton, which we mentioned in the previous videos, they form the adhesive bonds in a network that gives mechanical strength to tissues. Desmosomes are one of the stronger cell-to-cell adhesion types and are found in tissue that experience intense mechanical stress, such as cardiac muscle tissue, bladder tissue, gastrointestinal mucosa, and epithelia. Their skin. More info. Desmosomes are composed of desmosome intermediate filament complexes, DIFC, which is a network of cadherin proteins, linker proteins, keratin intermediate filaments. The DIFC can be broken into three regions: the extracellular core region or desmoglia, the outer dense plaque or ODP, and the inner dense plaque (IDP). The extracellular core region, approximately 34 nanometers in length, contains desmogliin and desmocolin, which are the cadherin family of cell adhesion proteins. Both have 5 extracellular domains and calcium binding motifs. Extracellular calcium helps form the cadherin adhesion by allowing the cadherin and extracellular domain on desmogliin and desmocolin to become rigid. They bind to each other via homophilic interactions in the extracellular space near their end termini. Desmogliin and desmocolin have a single pass transmembrane region plus an intracellular anchor to secure its position in the cell membrane. Desmogliins and the desmocolin A form contain intracellular cadherin domains binding to placoglobin. The outer dense plaque, about 15 to 20 nanometers, contains intracellular ends of desmocolin, desmogliin, and terminus site of desmoplakin. Armadillo family of mediatory proteins, placoglobin, placofilin, are involved in mediating attachment to intracellular filaments and cell membrane proteins. These consist of beta-carotene, P120 catenin, placoglobin, placofilins, etc. In desmosomes, placoglobin, placofilin help secure desmoplakin keratin filaments to desmosome structure. Placoglobin has 12 arm repeats with a head and tail structure. Placofilins have 9 arm repeats, exist in 2 isoforms, a shorter alpha and longer beta form. The inner dense plaque, about 15 to 20 nanometers, contains C-terminus end of desmoplakin and attachment to keratin intermediate filaments. Desmoplakin is the most abundant part of desmosome. It operates as the mediator between cadherin protein in the plasma membrane and keratin filaments. They have 2 isoforms differing in length of the middle rod domain. They all have an N-terminal head, C-terminal tails, 3 plakin repeats, a glycine serine arginine rich domain, GSR, at the C end. A picture connecting filaments, plasma membranes of 2 cells, cytoplasmic plaque, in the internal of the cells being attached, cytoskeletal filaments on the interior of the cells, and intercellular space between attached cells, cell membranes. Cell junctions are how cells are linked together. So we'll continue in the next video. Thank you guys. Bye. Say bye, Ruby.
Bye.